The function of Operon system is to regulate the gene expression in prokaryotes. All the organisms, include prokaryotes, need some mechanism to control the gene expression to save energy and resources. Expression of genes are tightly regulated based on when protein products are needed. Prokaryotes use operons to control the gene expression. Depending on the mechanism of function, there are two types of gene regulation or operons available. First of all, there is inducible or catabolic operon, where gene products usually function in the catabolic pathway. Lack of operon is an example of this catabolic operon. Another one is a repressible or anabolic operon, where gene products usually function in anabolic pathways. Tryptophan operon is an example of this repressible or anabolic operon. Now, what is Operon? Operon is operating units which can be defined as a cluster of genes located together in the chromosome and transcribed also together. It is a group of closely linked structural genes and associated control genes which regulate control elements which regulate metabolic activity. All the genes of Operon are coordinatedly controlled by a mechanism which first described in 1961 by Francis Jacob and Jack Enoch of the Pasteur Institute of Paris. Now, what is the basic structure of an operon? You can see there are basically three structural units in a single operon. The first of all, there is a series of structural genes whose expression is coordinated controlled by a single promoter and are operated, and there is also terminated sequence. In addition of this part of the operon, the key player for the expression of this structural gene is a regulated gene, which is located upstream of the operon. The promoter of operon is sequenced where RNA polymerase can bind and start the transcription of the structural gene downstream to the functional mRNA, and finally, which is translated to the functional proteins. In between the promoter and the structural genes, there is operator sequence. We can consider operator as a stop line for the RNA polymerase, either allowing or preventing RNA polymerase or transcribing all the structural genes. All of the structural genes in operon, a single promoter is there, as they all are involved in a simple metabolic process. The organization, this type of organization helps the system to be more energetically efficient. All the structural genes either expressed together or repressed together. Then there is a regulatory gene which encodes actually the repression proteins. This repression protein has DNA binding domain through which it can bind to the operator. Environmental factors interact with the repression and make it act or inhibit. When repression is active, it can bind. When repression is active, it binds to the operator and effectively prevents RNA polymerase to sit on the promoter. And no transcription of the structural gene will happen. When repression is inactive, its structure of the gene binding domain gets altered and the repression can't bind effectively to the operator. Then only RNA polymerase can effectively attach to the promoter and transcribe the structural gene altogether. Changes in the environmental factor alter the repression structure, so the functional proteins are only treated when they are needed. The most common example of the catabolic type of operon is the lag operon. Now we will describe about the lag operon. What is lag operon? Escherichia coli, when use lactose as a source of carbon, the enzyme required for the use of lactose as a carbon source are only synthesized when lactose is available as the sole carbon source. So, the lactose operon designated as lag operon variant The lac operon codes for enzymes involved in the catabolism or degradation of lactose. Lactose is the disaccharide which is made up of glucose and galactose. It is the inducible operon system since the presence of lactose induces the operon to switch on. What is the structure of the lac operon? The lactose operon consists of three structural genes. That is the lactate, which codes for galactosidase, an enzyme responsible for hydrolysis of lactose to galactose and glucose. Lac-Y, which encodes a galactoside farnese, which is responsible for lactose transport across the bacterial cell wall. And Lac-A, which encodes thiogalactoside transacetylase, which actually function of this enzyme is still, it is unknown. It is coded for by the enzyme by the gene Lac-A. Three structural genes are encoded in a single transcription unit, Lac-ZYA, which has a single promoter, that is the P-Lac. This organization means that three lactose operon structural proteins are expressed together as a polycystroline mRNA containing more than one coding region under the same regulated control. The Lac-ZYA transcription unit contains an operator site also, which is situated between bases -5 to +21 at the 5' end of the Lac promoter region. This site binds a protein called Lac repressor, which is a potent inhibitor of the transcription when it is bound to the operator. The Lac repressor is encoded by separate regulatory gene Lac-I, which is also a part of the lactose operon. Lac-I is situated upstream of the Lac-P-Lac. The Lac-I gene encodes the Lac repressor. The Lac-I gene encodes the Lac repressor, which is active as a tetramer of identical subunit. It has a very strong affinity for the Lac operator binding site, that is O-Lac, and also has a general high affinity for DNA. The Lac operator site consists of 28 base pair, which is palindromic. The inverted repeat symmetry of the operator matches the inherent symmetry of the Lac repressor, which is made up of 4 identical subunits. In the absence of lactose, the repressor occupies the operator binding site. So, in summary, we can see that there are 4 controlled elements in the Lac operon. First of all, there is an operator designated as Lac-O, which function as a binding site for repressor. Another one is a promoter site, Lac-P, which is act as a binding site for the RNA polymerase. The repressor protein gene encoding the Lac repressor protein binds to the DNA at the operator and blocks binding of the RNA polymerase at the promoter. And Lac-I controls production of the repressor protein. Now, how Lac operon works? First of all, again, the structure of the operon. The operon consists of the promoter, the operator site, and the structural genes that code for the protein. The operon is regulated by the product of regulatory gene I. Next, in general, the repressor is active and operon is off. This is the type of the catabolic operons. The repressor protein binds with the operator, preventing transcription from the operon. How the induction will happen? In the absence of an inducer, the Lac repressor always blocks, but a very low level of transcription of Lac-ZYA is there. When lactose is added to the cell, the low basal level of the farnese allows its uptake. And galactosidase catalyzes the conversion of some lactose to allolactose. Allolactose act as an inducer and bind to the lac repressor. This causes a change in the conformation of the repressor tetramer, reducing its affinity for the lac operator. The removal of the lac repressor from the operator site allows the polymerase, which is already sited at the adjacent promoter, to rapidly begin the transcription of the LAC-ZYA genes. Thus, addition of the lactose or a synthetic inducer such as isopropyl D thiogalactoside pyranoside, that is IPTG, very rapidly stimulates transcription of the lactose operon structural genes. The subsequent removal of the inducer leads to an almost immediate inhibition of this induced transcription. Since the free lac repression rapidly reoccupies the operator site and the LAC-ZYA transcript is extremely unstable. So, in general, the structural genes of the lac operon are controlled by negative regulation. That is, they are transcribed to produce an mRNA unless turned off by the LAC gene product. In fact, under normal conditions, the genes in the lac operon are turned off indeed. This is because it would be inefficient to produce enzyme when there is no lactose available. When cells are provided with lactose, the lac operon is induced, that is, the genes in the lac operon are turned on. The mechanism is that the few molecules of beta-galactosidase in the cell before induction convert the lactose to allolactose, which is an inducer. Another inducer of lac operon is IPTG, which I already told. However, the inducer IPTG is not metabolized by E. coli and is often induced for experimental studies of induction. Allolactose binds tightly to the repression and causes a conformational change that reduces the affinity of the repression for the operator site. The lac repression now dissociates from the operator site and allows the RNA polymerase to begin transcribing the LAC-Z, LAC-Y, and LAC-A genes. If the inducer is removed, the lac repression rapidly binds to the lac operator site and transcription is inhibited almost immediately. And in this way, actually, the lac operon is negatively regulated. Interestingly, the lac operon is actually subject to both negative and positive regulation. Cyclic adenosine monophosphate, cyclic AMP, is a single molecule whose prevalence is inversely proportional to that of the glucose. When glucose is absent, levels of cyclic AMP increase. Increased cyclic AMP levels mean that more cyclic AMP and catabolite activated protein complex can bind to a specific site near to the lac promoter and stimulate the transcription of structural genes. This protein, that is the catabolite activated protein, a dimer of identical subunits, which cannot bind to DNA unless it is complexed with the cyclic AMP. Thus, cyclic AMP and catabolite activated protein is also known as cyclic AMP receptor protein. So when glucose is absent, the levels of cyclic AMP in E. coli increases and the CRP binds to cyclic AMP. The CRP cyclic AMP complex bind to the lactose operon promoter PLAC just upstream from the site for the RNA polymerase. CRP binding induces a 90-degree bending in DNA, and this is believed to enhance RNA polymerase binding to the promoter, enhancing transcription by 50-fold. The CRP binding site is an inverted repeat. The CMP binding site is an inverted repeat and may be adjacent to the promoter, may lie within the promoter itself or may be much further upstream from the promoter. The differences in the CRP binding sites of the promoter of different catabolic operons may mediate different levels of response of these operons to cyclic AMP in vivo. Thank you.